I'm an entomologist and evolutionary biologist at the California Academy of Sciences, and even though it's really my job to professionally love insects, which I do, there are some that I definitely prefer more than others. So I did just move from the East Coast pretty recently, and these guys were a relatively common roommate of mine. I don't know, have any of you guys encountered these enormous, repulsive cockroaches in your life before? So it's so great that I haven't, yes, some. I haven't seen any out here on the West Coast yet, which is nice. But again, even though I'm an entomologist, if I encounter one of these, I probably will flail and shriek and it'd just be an embarrassment to the entomological community. But one of the things I really love about insects and arthropods, which are all kind of creepy crawlies, is the fact that they provide this really great reality check about our place in the world. And so as humans, I think particularly in Western society, we really see ourselves as set apart from nature, right? We build these islands kind of that we live on and we separate ourselves. You know, we can partake in nature when we want, but then we sort of also get to opt out. But to some degree, that couldn't be farther than the truth, right? When you think about the wild world, we're really right in the thick of it. So even our bodies are these incredible conglomerates of microbes and fungi and viruses, you know, and I'm sure you all were as shocked as I was to hear, you know, a few years ago that it's possible that more cells in our body actually belong to other species. Like most of our cells aren't even Homo sapien, which is just crazy to think about. And it's not just the microbes that keep us connected to the natural world, it's also the arthropods. And again, not all of them are my favorites. These are some troublemakers. But arthropods have been living and evolving on the planet for about 500 million years, and so they've been with us since our very earliest origins. We haven't ever really been able to escape their impact. And what I really appreciate is that arthropods have no regard for our desire to be set apart, right? They completely disrespect our otherness. They have no patience for our snotty attitudes. And basically to the arthropods, we're really just an extension of the habitat, right? We're part of the ecosystem. Our houses, our bodies, our food are all, you know, the arthropods think they're all fair game. And so again, a lot of these arthropods we'd be happy to do without in our regular life, but today I'm not going to focus on any of these kind of more conspicuous troublemakers. I'm going to talk about my very favorite group of arthropods that are invisible and quiet and introverted and lovely. They're like, you know, beautiful wallflowers in the arthropod world, and that's our face mites. So this is a very distinguished portrait of a face mite. If you've seen a face mite picture before, you probably haven't seen one like this. We're looking kind of at his chin. Like if he wore a tie, it would go right here. This is the kind of chest of the face mite. So it's a very dignified pose. So here is a little video. Hold on, I just want to show you this thing creeping around a little bit. This was just made with a cell phone in our microscope. Oh my goodness. Every time I see him this big on a screen moving around like that, I just get so excited. So here's his head and his stubby little legs. He's got this long tail that belongs right deep in your pore. So for me, my face mite research was really born out of this complete shock and awe that we have arachnids living on our face, right? I mean, even now I've been studying them for several years and I can't get over it. Like if I really think about it, I'm newly shocked and in awe about the fact that these you know, spider relatives are living on our face right now. And so these mites are called face mites, but actually they live in various parts of our bodies, so they could equally be called nipple mites or genital mites. I'm hoping to shock you more. And one might expect that because they're living on our face that we'd know a lot about them, right? But unbelievably, we know very, very little about them. In fact, there's been such a small amount of molecular work done on these mites that it's really shocking for me. And this really triggers in me kind of my middle school science brain. I remember sitting in my science classes, and probably you guys do too, and I just had this sense then that like all the cool stuff was already figured out. Like all the big discoveries, all the fun stuff, like we already knew all that. And so for me, when I realized as an adult scientist, like, oh my gosh, we have arachnids living on our face and we know almost nothing about them, like how is that possible? So that really gets me excited about making you know, big new discoveries. So like I said, there has been very little molecular work done on these mites, but now that I've worked on them, I totally get it because they're actually a huge pain to work on. They're really hard to study. So here's what we know about these mites so far. They're from the genus Demodex, and so all of them are microscopic, and they're actually part of our mammalian heritage, so all mammals have these mites. They live in our skin. There's about 150 described species, and they've been found on every mammal that they've been examined for, except for the platypus and its weird egg-laying relatives. And so often, a mammal has more than one species. There's this one poor field mouse that has like 4 different species, all living in different microhabitats all over its face. So these mites have really been able to exploit mammal skin environments. Now, it's pretty crazy because there's about 150 described species of these things, but there's well over 5,000 species of mammals. And so if you imagine that each mammal has probably conservatively 2 mite species, that's well over 10,000 mites that we have yet to discover. And scientifically, it'd be really exciting because if you think about mammals, we have this really well-resolved mammal tree of life. We know how mammals all split off from each other and when. And if we could build that same kind of tree for Demodex, it would provide this incredible comparison of kind of host shifting and coevolution and microhabitat specialization. So for me, that's an exciting scientific goal, although right now we're a long way away from that. So back to people, we have less than that lucky field mouse. We have 2 species of Demodex that live on us. The first one, Demodex folliculorum, is this long, skinny one over here. And it's the more common and gregarious mite. It likes to live more superficially in your pores, and it likes to live in groups. Then next to it is its short and stubby cousin, brevis, and that lives deeper within your sebaceous glands. And so interestingly enough, just because one's long and skinny and one's short and stubby, you can't actually necessarily identify them that way. So it turns out you really have to use DNA to know exactly what species of mite you have. So there's a lot of, again, kind of cryptic diversity and discovery that we can make with molecular data. Okay, so this was a National Geographic video. I'm having some audio trouble, so I'm not going to be able to play it for you. But National Geographic made a little bit of a short clip about our research last summer. And so this was a video of Dan Fergus, who's the molecular biologist that I collaborate with on this project. And he's basically talking about the life cycle of the mite. And what he says is that mites live for about 2 weeks on your face. And again, the natural history is not really well known. The life history is not really well known. So a lot of this could just be storytelling. But apparently the mites are deep in your pores until nighttime. At night, they like to come out and crawl around your face. Now, not very far, because you saw those legs, right? They're really stubby. They crawl around your face and have sex on your face. Then they go back into the pores. Now, another funny thing about these mites is that apparently they have no anus. So again, not lunch appropriate, but they have no anus. And so people have suggested that they just eat and eat until they finally one day explode. Now, we think that's probably not true because we've looked at a lot of mites. We've never seen like an exploded one or like an engorged one. And apparently some mite researchers say, plenty of mites don't have an anus. Who knew? But so again, a lot to learn about these mites. Okay, so when I started this project, it was mostly an outreach tool. I worked in a science museum in North Carolina, had a glass-walled lab, and I was really trying to engage the public in science and the diversity of life on your face. So we just basically wanted to show people their mites. But apparently finding live mites on people isn't always easy. So there's a handful of ways to get them off, and I've tried all of them. You can put tape on your head and then peel it off. They say it's better if you do that at night because that's when they supposedly come out. You can pluck your eyebrows or eyelashes, and sometimes if you look under a scope, you find mites at the base of the hairs. But I was doing this with the public, and the pain to gain ratio wasn't really worth it, plucking all these people's eyelashes. Or you can scrape your face with a tiny kind of spatula, which is what we often do, along your nasolabial fold, which is an area of your face I never knew how to name, but right down here apparently is a very mighty place on your face. So along this whole process of the past few years, I've gotten to scrape some VIPs. This is Ed Wilson. He's a celebrity in my world, maybe not in yours, but maybe you know him as E.O. Wilson. He's a famous writer about science and ants. He had no mites, which was a huge disappointment. Okay, so the most effective way of all, which you would have gotten to see in the little video, is sounds a little bit rough, but basically you put super glue on a slide, stick the slide to your face, wait for about 30 seconds, and then pull it off. Apparently, this is like a standard biopsy method for dermatologists. I had no idea, but it's not as bad as it sounds. I've done it many times to myself. And actually, for about the first 9 months of this project, we knew from our molecular work that mites were pretty common, but I couldn't find any mites on myself. And I did all the methods. I was just always looking for mites. And so I kind of had a little bit of a superiority complex. Like I thought, all my colleagues, they're so gross, they're covered in mites, but I don't have any. Then finally one day I did the Superglue Method, and I pulled off like 20 mites the first time I did it. So this is actually a great way to get mites. And actually I have to say I was kind of relieved, because as much as I felt really good about myself not having any mites, I was also a little bit terrified. Like, what kind of toxic environment have I created in my body that I've killed my whole natural mite population? So initially when we started sampling the public, our return rate was pretty low, meaning we just didn't find mites very often. But we did it so much, hold on. This is another little video. We did it so much that we really got to see a lot of cool biology. Oh, here we are. And I'm just going to show you something that we videotaped. Are you going to work? Oh, maybe I've broken the whole thing now. Okay. So watch this mite. Aww. Okay. Gonna move it along. Oh my gosh, we were just watching this under the microscope. Look at this. So we had no idea what was happening. And then we compared it to some pictures in the literature, and what we realized is that this mite was laying an egg. Look at this, it's an enormous egg. It's like a third of the size of its body. And it has this really distinctive boomerang shape, and so we recognized it immediately once we saw the other picture. But oh my gosh, so I don't know how many people have gotten to see a face mite lay an egg, but for us, this was like a really big deal. And in fact, one of my colleagues, he's a science comedian, he is like the mightiest person I know, and so I always am scraping his face for mites. And he got to watch this, and he is so proud. He calls that his grandbaby, that egg right there. He's so happy about it. So again, our return rate was really low, and so we were only finding mites on about 10% of the people, and it took hours. It's a lot of work to scrape faces, look under a scope, find a mite, then try to manhandle that tiny mite into a little vial. It was too much work. And so things were moving really slowly. And then we had this incredible breakthrough. Again, my friend Dan Fergus, a molecular biologist, he just decided, we scrape everyone's face and we get all this gunk off their face. Why don't we just sequence that for mite DNA and see what we get? Maybe we don't have to actually have the body of a mite to get mite DNA. And so this seems really obvious in retrospect, but it was a big eureka moment for us, because it basically meant we could get molecular data from mites without having to go through all that rigmarole of trying to manhandle that little microscopic mite. And so this really helped push our research forward. So I'm going to tell you the 3 most important things that I've learned about face mites now in the past couple years since I started this project. And the first thing is that you have them. I hate to break the news, but based on our experiments looking at presence, absence of mite DNA off of scrapings from people's face, we found that everyone, 100% of people over the age of 18, had evidence of mites on their face. And so that makes us think that none of us are immune. They're completely ubiquitous in the human population. And again, this is really in stark contrast to the 10 to 25% of people that we would find mites on when we were looking for actual live bodies. And so a lot of the literature reports this rate that, okay, maybe 10 to 50% of people have these mites. But there was a lot of variation, because if you looked at dead bodies, they would find them on, again, almost everybody. And I think it's just because they're hard to find. They're hard to actually get off of your face. But it doesn't mean they're not there. And so we also found that face mites are really your own. Okay, so you're not casually sharing these mites here as you sit at the lunch table, or when you kiss cheeks hello, or these mites live deep in your pores, and like I showed you, they've got stubby little legs. They're not going very far very fast. And so I think that you probably get colonized by your mother's mites when you're young, and you probably keep them for the rest of your life. And now you do share mites with your partners and your kids. Parents and offspring share mites, but again, you're not sharing them casually. So the next big exciting finding for me is that these 2 species that live on our face, brevis, the short tubby one, and folliculorum, that long skinny mite, are incredibly distant cousins. And that opens up this whole can of questions. When and where did we get these mites? How did we get these mites? How did they come to live on us? And so by comparing the mitochondrial genomes of both brevis and folliculorum, it's been shown that they probably diverged from each other, well, many, many millions of years ago, up to 80 million years ago, which, for perspective, humans are probably 200,000 years old. Our genus Homo is 2 to 3 million years old. So we're talking 80 million years. Like, these mites are so different from each other. It's like a pig and a bat or something. Now, I haven't checked those dates, so don't quote me on that, but I'm just saying they're very, very different from each other. Yet, you can see they look pretty similar. They both live on our face. It's very, very curious. So they probably colonized the ancestor of humans before mammals ever even evolved. And so where did we get these mites? And again, your guess is as good as mine, but it seems like these mites have been with us for all of our history, as early, again, as the origin of mammals. So before mammals split off into their big order-level divergences, like before rodents split off from pigs and bats and primates. And so it seems like these mites had the ability to really adapt to new skin environments and then speciate with mammals. And so I would predict that we inherited one, if not both, I think probably both of our species of mites from our primate ancestors. So they've been with us for a long, long time. And so then as ape and other hominid species evolved, our mites just evolved with us. And so if that was the case, we'd expect that chimpanzee mites would be the closest relatives to our face mites. And so that's something that I really want to know. And unfortunately, it's incredibly difficult to get mites from a chimpanzee. So I've been able to get some swabbings of a chimpanzee from a zoo, but I really need wild chimpanzees. But I just got an email yesterday from a chimpanzee biologist that said, you're never going to get that. No one's ever going to be scraping mites from a wild chimpanzee. So apparently that's going to be like, the dark void in science for a long time. Okay, but my very, very favorite part about my face mite research is the fact that I've uncovered the fact that face mites can tell us about our own evolutionary history. So they're not just bugs on our face, they're really these incredible storytellers. And so looking to parasites to understand our own evolution isn't new. So as humans have evolved and moved around the world, we take this whole suite of species with us. And most of what we know about human evolution comes from fossils or our own genetic data. For example, like the recent sequencing of the Neanderthal genome that's shown us how we've really intermingled with other Homo species, which is so cool to think about. But you may not have realized that other arthropod parasites have also told us a lot about our evolutionary history. My favorite work is on lice, and it comes out of a lab in Florida. And they've done the most amazing studies. Now, again, I have little kids, and lice are a huge pain. Like, I'm not a huge fan of lice, except when it comes to science, because the lice research has revealed amazing stuff. So basically, they've showed us when humans have lost their hair. They've been able to date when humans lost their hair based on lice research. They've been able to date when humans started wearing clothes by looking at body lice that live in clothes and their evolution. And they've also provided independent confirmation that we also intermingled with other Homo species, just like we got from the Neanderthal genome. More disturbingly, we found out that we also probably intermingled with gorillas because that's where we got our pubic lice. Bizarre. And so now I'm still in the really early days of this research, but we're finding that different groups of people host different mite lineages. So Asian and African face mites are different from European face mites. And what's amazing about this is that we find, for example, Asian face mites on Asians who've been in the US for decades. And we find African face mites on African Americans who've been in the US for generations. And so this is just how graphically I display my data. And these are two trees showing the genetic divergence in the mitochondria of folliculorum and of brevis. And so what's cool about it is you can see these super long branches belong to people of African, or come from mites from African hosts. And that's the same in both folliculorum and in brevis. And so basically that the really long branches, these big, deep divergences are in African hosts and Asian hosts. And that represents kind of the same pattern that we see in human genetic diversity, right? Because we're all from Africa, most of our genetic diversity is in Africa. And so these mite lineages appear to reflect our own ancient migratory history. Again, the oldest lineages are found in people from Africa and Asia. And then this is This is a graph from National Geographic Genographic Project, so looking at the mitochondrial DNA of humans all over the world to try to recreate our migratory history. And basically, what my mite tree that I just showed you sort of reflects is the fact that there's these long branches of mites that evolved here in Africa, and then there's this split that happens. Then we have these Asian mites that also have deep, long branches. Then there's a split that happens between Asians and Europeans, and that perhaps happened about 40,000 years ago. And so maybe we'd expect that that long branch represents that 40,000-year divergence between European populations and Asian populations. So just to clarify that again really quickly, both of these trees have this enormous burst, and that's the kind of the area of the mite tree that has very limited genetic diversity, and it's found in mites that are primarily on Europeans. So Europeans occur in no other part of the tree. So it almost looks like there's this recent population expansion that's happening in mites from Europeans, and it almost represents like global colonization. So like these European mites can live on all types of hosts, people from Africa, Asia, Latin America, and this population is expanding. So again, this is just the beginning of the story, but it seems like we have this really exciting line of questioning to follow. So now, I just pretty recently moved to the Cal Academy over the past year, and I want to expand this mite project, along with another project I have done to explore the diversity of arthropods in homes, into this global survey of the diversity and evolution of arthropods that are part of our daily lives. And so, when you think about the unexplored ecosystems in the world, I think we often think about like rainforests or the bottom of the ocean. But the truth is that some of these places that are closest to us, right, our houses, our bodies are also still really unexplored ecosystems. And we can still make big discoveries in these really familiar places. And so I'm launching this project I'm calling the 7-Continent Study, although I'm thinking of calling it the 6-Continent Study because apparently getting to Antarctica is really hard and expensive. But anyways, it's going to be consist of expeditions to each of the 7 continents, focusing on sites that are unique in terms of human history and biogeography. And along each way, I'm going to sample face mites on people and sample arthropods in houses in both urban and traditional settings. And hopefully I'm going to uncover more of this ancient story of arthropod evolution with humans and how they've adapted to humans over recent time, and to see how that varies in different parts of the world. And for me, the most interesting part is thinking about how do our face mites vary across this incredible, beautiful gradient of human diversity. And I'm getting a glimpse of that right now, but there's still a lot more to come. And you can see I included Barack Obama here because when I think about his face mites, I just can't figure it out. He has an African father, a European mother, but then he lived in Hawaii and the Philippines, so who knows? Who knows where his face mites are from? He's like going to be as hard as getting the chimp mite, right? I probably am not going to get Obama's face mites. Anyways, that's all I have today. A big thanks to This Is My Mite team who helped make this project happen. And thank you very much for letting me talk to you about bugs while you're eating your lunch. Sure, I'm free for questions if, yeah?
Yeah, I thought it was interesting that face mites just stick with you, like their environment.
Yeah.
Is it more common for arthropods? Because I know lice, move around. Is it more common for arthropods to stick with an animal or to just be agnostic?
No, I think that's why they're unique and they're a really potentially powerful system because lice, for example, like you said, there's a lot of what you want to consider horizontal transfer, right? They'll move between people, but these face mites really are more like your own genes in the sense that they are transmitted primarily vertically. I mean, some horizontal between spouses but really a lot of vertical transmission, parent to offspring. So that's unique.
Is there any others that they provide any benefit to humans?
She asked if there's any benefit to humans. And so my standard answer to this is the fact that, again, we've learned so much about the importance of the microbiome, our gut microbiome, our skin microbiome over the last decade. And so these mites have been part of our kind of skin ecosystem for such a long time. I mean, obviously since our origins. And so because of that, I think they must contribute to the healthy skin environment. Again, benefits, I don't know, but it might be a stretch. But they certainly are a natural part of our skin environment. I'm going to Google that. I don't know. I think, I'm sure you could locally kill a lot of face mites. I don't know if it would make your pores smaller. I don't know. They're so small. I don't know if they're really doing a lot to, and they're kind of feeding at the bottom of your pores, so I wouldn't expect they'd play a big role in making your pores bigger. But I would say if you killed all your face mites on your face, they would probably recolonize from your ears or from some other part of your body or from, your partner. So I wouldn't, and also, kill all of them. Like all you need is like one egg left in there. It's like trying to get rid of head lice, right? Like, you have 2 eggs left somewhere and then they're back again. So it's hard to stop arthropods. Like they will find a way, right? So I would be skeptical. I wouldn't pay a lot for that. Well, and I didn't mention this before, they have been associated with rosacea, like a skin disorder. I know someone who has kind of like a weird chronic eye infection from them, and it seems like some people might have an immune response to these mites. Because they're on everyone, clearly they're benign most of the time, and I think, again, contributing to the healthy skin environment. But it seems like some people do have some kind of immune response. Another sign of that is that dogs sometimes get mange from these mites, but it's not a contagious mange. So again, it seems like it's limited to their own immune reaction.
What prevents other kinds of species of mites taking over your body?
I don't know.
How do they fight off?
I don't know. So again, we have these 2 species, so there must be some competition going on there. They have themselves in sort of different microhabitats. But more interestingly is, so we have these major lineages of mites. And again, people with African ancestry often host African mites, but Europeans never host African mites, never. Now, Africans can host European mites, but Europeans can never host African mites. And so what's constraining that? I don't know. I need an immunologist or a dermatologist, if anyone knows and wants to collaborate with me. But whatever it is, I'm sure it's interesting. It's fascinating.
Going into this 7-continent study, do you have a particular hypothesis?
Well, so for me, one of the driving questions is to first just get the samples of human face mite diversity. And what I think is that if you go back to this tree, you can see how the Europeans are really kind of like this explosive bush here. And I think if we did a lot of sampling in Africa, the tree would be more similar in those regards, right? There's so much African diversity and Asian diversity. And I also think that potentially I'll have the ability to answer some questions about human migration, and so that would be really exciting to me. So that, for the face mite piece, for the arthropods and houses piece, my driving motivation is really to get just a very foundational picture of what types of arthropods live in human houses and how they've potentially adapted to human houses. And we have such little information about that again, it's just kind of very basic characterization. But what I want to get out of that is a core community of arthropods that have really evolved with humans over the last 20,000 years. Like humans have only been building houses for about 20,000 years, so.
Could you get face mites off a dead wild chimp?
Oh yeah, I need can you get me a dead wild chimp? Yes, I think you could. And in fact, I mean, we have collections. I don't think we have a chimp in our collection, but like going to museum collections might be another route. I'm really stuck at this chimp thing. I need to figure out a way to get my hands on some chimps.
How long can mites live away from their host?
So not long, and that's been another problem for developing more of the science about these mites because they basically can't. So we can't culture them. There's a bunch of papers, not a bunch, but a handful of papers about people who tried to culture these mites. And like one person had hamster skin folded in between chicken skin where they were trying to grow these mites. I mean, just bizarre stuff. And I think the most people have ever kept them for is, a handful of days. They just can't seem to be kept off of a host.
But you mentioned that you get them off dead bodies. How long do they stay on dead bodies?
Yeah, well, I don't think you can get them live off dead bodies, but they find their bodies on dead bodies, yeah. Because they have an exoskeleton, they can kind of maintain their integrity for a while on dead bodies. On that note, it's a positive note to end on. Thank you again very much.